Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Jared Isaac, PhD, Associate Director, Chromatography and Mass Spectrometry from Cygnus Technologies.
Today I will talk about advanced orthogonal methods to fully characterize process host cell proteins. Why do we use ELISA to quantify host cell proteins? ELISA is the gold standard for process monitoring and product lot release, primarily because it can measure nanogram per mil levels of host cell protein and the presence of mg per mil level of product protein. However, it has its pros and cons as shown here. We know that ELISA is a semi-quantitative measurement of a statistical sample of the host cell proteins in a given sample. If designed well, it can have excellent sensitivity and selectivity. ELISA does not require special expertise to run, and it can be transferred across different departments, companies, and manufacturing processes internationally because it is reliable and robust. One of the cons of ELISA is that it reports the total host cell proteins. Individual host cell proteins are not identified or quantified. And while host cell protein ELISA does not require a high level expertise to run, it, however, does require a high level expertise to develop a broadly reactive host cell protein antibody and then turn that into an accurate and robust assay. Finally, orthogonal methods are required to assess the ability of a host cell protein antibody to recognize a wide range of host cell proteins that are present in each bioprocess. And ELISA may be qualified as fit for purpose of process monitoring and product lot release. These are the criteria that need to be established or passed before an ELISA can be used to monitor your process. You must first conduct dilutional linearity, accuracy, precision, and then determine the range of your assay. This includes determining the limit of detection, the lower limit of quantification, and the upper limit of quantification. With those tests passed, the next thing you would do is determine antibody coverage, and this can be determined by antibody affinity extraction coupled with 2D page and silver stain detection. If available, you can also characterize your assay in more detail by performing antibody affinity extraction coupled with mass spectrometry. The intent of ELISA is to quantify host cell proteins that may be potentially problematic. We know from the literature and from Cygnus Technologies' over 25 years of host cell protein experience, there are many host cell proteins that affect drug product stability. Host cell proteins can affect drugs by degradation through protease activity of the drug itself, promoting drug aggregation, degrading PS20 and PS80 drug stabilizers, increasing the immunogenicity of the drug, and causing dilutional linearity issues. Some of these proteins you may be familiar with on this list. A very familiar protein on this list to many of you is maybe phospholipase B-like 2. As you can see from this 2D virtual gel, the host cell proteins on this list have isoelectric points and molecular weights of different ranges. This is unfortunate because these ranges are also shared by drug products such as recombinant proteins and monoclonal antibodies. Next, I'll go into more detail about methods for determining antibody coverage. The first step you need to do to demonstrate that a host cell protein ELISA is fit for purpose is to demonstrate that the antisera used in the ELISA is broadly reactive to the array of host cell proteins that are found in each bioprocess. Traditionally, the regulatory expectation for the coverage assessment has been a large format 2D gel, or actual 2D Western blot comparison to a silver stain. In this method, a sample is run on duplicate large format SDS gels. The proteins from one gel are transferred to a membrane and detected using the anti-host cell protein antibodies by Western blotting. The duplicate gel is then silver stained. The percent antibody coverage is determined by aligning the two images and determining the number of spots detected by silver stain that have a corresponding Western blot reactive spot. The broadly reactive antibody must react to a majority or greater than 50% of the total host cell proteins, recognized proteins, in all quadrants of a gel. In the second part of my presentation, I will show why it is a poor practice to use and to rely on 2D Western blot to determine host cell protein antibody coverage. To overcome the limitations of 2D Western blot, Cygnus Technologies developed antibody affinity extraction, or AAE, in 2014. AAE is referred to as immunoaffinity chromatography in the USP Chapter 1132. This is a workflow of how you can analyze harvest material by antibody affinity extraction to determine antibody coverage. An aliquot of a harvest, CCF, or cell lysate sample is saved. To perform AAE, the polyclonal ELISA antibody is covalently immobilized on a chromatography support. The column is then conditioned to prevent significant leaching of the antibody and to minimize any nonspecific binding. The remainder of the sample containing host cell protein sample in its native, undenatured state is passed over the column for binding and then eluted with acid. The host cell protein sample is then cycled over the column by binding and elution until no additional host cell protein is bound. All host cell protein elution fractions are pooled, buffer exchanged, and concentrated back to the original sample volume. The resulting sample represents all the immunoreactive host cell proteins captured with the immobilized polyclonal ELISA antibodies. Next, we have two options for the analysis of the samples. Option 1 is 2D page of silver stain or fluorescent staining, and option 2 is LC-MS. These results look different based upon the analysis. For option 1, there are gel images, and a percent coverage is calculated. In option 2, we have a virtual 2D page image, percent antibody coverage, and a list of all the host cell proteins in the sample, as well as the immunoreactive host cell proteins with their molecular weight and isoelectric point. Here we show a 2D page workflow for host cell protein analysis. We have combined AAE with fast protein liquid chromatography. We then perform sample preparation, isoelectric focusing, 2D page, and silver staining. Gels are then imaged and spots are counted using bioinformatics software with scientist data analysis to calculate the antibody coverage range. In mass spectrometry-based host cell protein analytics, each sample type is a unique consideration. Instrumentation that I'm showing on this slide are intended to be illustrative of the potential combinations that may be employed for host cell protein analytics. In general, samples are enriched by for host cell proteins by AAE and tryptic digested. Custom LC-MS methods are then developed to the specific host cell line, drug substance, sample matrix, and purification process. After method development, samples are run by liquid chromatography mass spectrometry upon a high-resolution accurate mass spectrometer, such as an Orbitrap-based instrument as shown here. Mass spectrometry data are searched using bioinformatics software. All data searches use Cygnus Technologies' proprietary curated proteomic database that has been developed and curated with common species expression systems and potentially problematic host cell proteins. Here I present the specificity assessment of the AAE method using a CHO harvest sample. In panel A in the upper left, a CHO harvest sample was run over the F550-1 CHO 3G AAE column. 62% of the CHO sample was bound and then eluted from the specific CHO 3G antibodies. For the non-immune goat IgG control, a negligible amount of the protein was bound. In panel B, we have the AAE mass balance experiment of one pass over the AAE column. A complete AAE run includes 4 passes over the AAE column. Going from left to right, the starting sample was flowed over the AAE column. Of the initial 100% initial protein content, 88% of the protein was contributed to flow-through, 8% was bound and eluted, and 4% of the total protein was in the dead volume of the FPLC. In the upper right, we have panel C, and we have the FPLC chromatogram of the CHO antigen with the anti-CHO host cell protein polyclonal antibodies. As you can see, each pass over the AE column, a specific elution peak is generated, which decreases with each subsequent pass. This is done a total of 4 times. In panel D, we have the AAE of the CHO antigen with non-immune goat IgG. However, with the same CHO antigen load, there is no elution peak with a non-immune goat IgG. Taken together, these data support the specificity of the AAE method. The second part of the AAE method specificity was assessed using an HEK harvest sample. AAE was performed with the F650S antibodies, and 2 detection methods were used: 2D page and mass spectrometry. In panel A, we have the pre and post AAE 2D page gels. In panel B, we have the 2D virtual gel of the MS data with matching proteins in green, non-immunoreactive proteins in red, and enriched proteins in black. Enriched proteins are represented by black dots, and they were in the pre-AAE sample as well, but they were below the method limit of detection. As you can see in the table below, the 2D page detected 1,687 spots in the pre-AE fraction and 1,604 spots in the post-AE fraction. The antibody coverage by 2D page detection was 97%. With mass spectrometry as a detection method, 3,075 proteins were identified in the pre-AE fraction, and 2,812 proteins were identified in the post-AE fraction. The antibody coverage by MS detection was 91%. As you can see, these 2 methods are in close agreement with each other, and mass spectrometry detection is a more conservative detection methodology for coverage as compared to 2D page. We compared available antibody detection methods and determined AA 2D page and MS are superior to detection methodologies. In this example, antibody coverage was performed on a SF9 null harvest sample using the F840 SF9 antibodies from Cygnus Technologies. Detection methodologies were used to determine antibody coverage to the harvest sample. Starting from the left, 2D page determined the antibody coverage to be 94%. Moving to the right, 2D Dig Western reported that coverage was 88%, AEMS reported coverage as 72%, and ELISA MS had the lowest antibody coverage percentage of 60%. The differences between AEMS and ELISA MS are the reason that AEMS outperforms ELISA MS. AAE extracts microgram quantities of protein to be analyzed for deep and sensitive identification of host cell proteins. In comparison, ELISA MS can only extract low nanogram to picogram quantities of host cell proteins from the sample ELISA well. This leads to less sensitivity and lower detection of host cell proteins due to their lower abundance. AAE empowers low-level host cell protein identification and accurate antibody coverage assessments. Thus, AEMS does not overestimate antibody coverage. ELISA polyclonal antibody coverage is highly dependent upon detection method. In this example, the same sample was analyzed by Cygnus Technologies antibodies and detected with different detection methodologies. The different methods used were 2D Western blotting, antibody affinity extraction of 2D page, and AAE with mass spectrometry detection. As you can see in 2D Western blotting and AAE 2D page, the same number of pre-AAE spots were detected. However, in Western blotting, fewer spots were detected compared to AAE 2D page. This leads to the difference in the coverage ranges between 2D Western blotting and AAE 2D page. Furthermore, in AEMS, we see the highest coverage range represented, and we can identify even more pre-AE proteins and a higher number of post-AE proteins. AAMS is a highly sensitive detection method for determining ELISA antibody coverage. This shows that for the same antibody, the coverage percentage is highly dependent upon the methodology used to assess the coverage. Next, I will talk about using 2D Western blot methods to assess antibody coverage. This is a representative workflow for 2D Western blotting. The sample is prepared by a proprietary protocol and split into 2 samples. Next, the samples are separated by isoelectric point and then separated in the second dimension by molecular weight. However, this is where the difference happens. The first gel is visualized by an in-gel stain. The second gel is transferred to a membrane. The protein that has been transferred to the membrane is then probed with an anti-host cell protein antibody and detected typically by a new conjugated second antibody. Images are then collected of the in-gel stained proteins and detected membrane proteins. Coverage analysis is calculated by software of the two images. For a lack of better methods, 2D Western blot has been traditionally performed from 2D page with protein transfer to membranes for 2D Western blot comparison to silver stain. The in-depth discussion in the data review demonstrating the limitations of 2D Western blot for host cell protein coverage analysis or detection of host cell protein that could potentially co-purify a drug substance is outside the scope of this particular presentation. However, here is a quick review of the data showing that the same antibody that interacted with 73% of CHO host cell proteins from the previous slide demonstrated 55% coverage based upon the 2D Western blot results from the image on the left. Moreover, when the CHO host cell proteins were probed with non-immune goat Ig, 48% of the host cell proteins were detected in the image on the right. These results clearly demonstrate that when using 2D Western blot, you can significantly underestimate the true antibody coverage to upstream host cell proteins, in addition to having poor specificity. For these reasons, we do not recommend relying on 2D Western blot for establishing host cell protein antibody coverage, especially as more advanced orthogonal methods are available. This is an example of the results of coverage analysis by AAE. The image on the left is the starting clarified harvest material and represents all the host cell proteins that could potentially find their way into the purification process. The image on the right is a silver stain of the host cell proteins found in the AAE elution fraction. This represents all the host cell proteins that the antibody specifically interacts with. Image analysis showed that 827 of the 1,138 total spots were found in the AAE elution fraction for a coverage of 73%. In this example, the Cygnus Technologies third generation CHO host cell protein antibody was used to construct the AAE column. AAE is a powerful method in completing the host cell protein picture. It has superior sensitivity and specificity compared to Western blotting. AAE can be used to perform antibody coverage to demonstrate that the ELISA is fit for purpose, as well as analyze host cell proteins that persist through the purification process. ELISA polyclonal antibody coverage is dependent on the detection method. 2D Western blotting typically has a coverage range of 50 to 60%. AAE combined with 2D page in general has a coverage range of 60 to 85%, which is also dependent on the staining methodology. If you use silver staining, you can typically get a coverage range of 60 to 80%. Fluorescent staining has a coverage of 70 to 80%. And Psi3 and Psi5 dyes have a coverage range of 70 to 85%. However, combining AAE and mass spectrometry, we can obtain a coverage range of around 70 to 90%. So I spent a lot of time talking about coverage ranges, but how do we determine ELISA polyclonal antibody coverage? We use ranges, and that range is developed by using a lower and upper boundary to obtain that antibody coverage range. The lower boundary is calculated by the post-AE spots divided by the number of unique spots. The upper boundary is calculated by the post-AE spots divided by the pre-AE spots. This is a more direct coverage calculation. As you can see, the difference between gel and mass spectrometry-based detection is that we're detecting visual spots that are a qualitative representation of proteins versus mass spectra, which represent quantitative ions. For the next section of my presentation, I will present case studies where AAE and mass spectrometry were used to determine host cell protein antibody coverage and identify and quantify host cell proteins in process samples. AAE-MS was used to determine the antibody coverage of the CHO3G F550-1 antibodies. In the top left of the slide, we see a virtual 2D page gel image of the F550-1 antibodies with the CHO antigen sample. 1,673 CHO host cell proteins were detected in the pre-AE fraction, and the post-AE fraction, 1,639 proteins were identified. Host cell proteins that are immunoreactive with the F550-1 antibodies are green spots. Host cell proteins that are non-immunoreactive are red spots. Enriched host cell proteins in the post-AE fraction are black spots. These numbers lead to a calculation of antibody coverage of 97%. On the right side of the slide, we see a list of the top 40 potentially problematic CHO host cell proteins in the CHO antigen sample. This table contains the names of these top 40 CHO host cell proteins along with their molecular weight and their isoelectric point. As you can see, the majority of the host cell proteins are detected in green. Undetected host cell proteins are shown in red. Most importantly, this table tells us that for the potentially problematic CHO host cell proteins in the CHO antigen sample, both the F550 and the F550-1 antibodies are immunoreactive with them. Another important advantage of using AEMS for coverage analysis is that we can use product-expressing harvest material instead of null or mock transfected harvest. This is important for two reasons. First, our clients may not have easy access to nil cell lines, and this happens more and more often. Secondly, product-expressing harvest is a better representation of all the host cell proteins that are secreted into the media together with a monoclonal antibody during production runs, and thus this provides more accurate coverage assessment. Ideally, once the selected host cell protein lysis method is qualified and validated as fit for its intended use of process monitoring and product lot release, it is imperative to ensure that critical reagents supporting the validated host cell protein lysis can last throughout the lifetime of the program, from supporting phase 1 to phase 3 clinical trials and post-market product life cycles. However, since ELISA polyclonal host cell protein antibodies cannot be supplied indefinitely, sometimes these critical reagents must be resupplied. In a recent example, Cygnus Technologies has transitioned the depleted CHO host cell protein antibody 3G and the corresponding CHO host cell protein lysis 3G from the original F550 kit to F550-1 kit. AEMS was chosen to bridge the F550 CHO3G to the F550-1 antibodies. On the top left, we have the virtual 2D gel of the F550 antibodies. On the top right, we have the virtual gel of the F550-1 antibodies. The table below summarizes the F550 and the F550-1 antibody coverage. 1,673 CHO host cell proteins were identified in the CHO antigen sample. Of those proteins, 1,649 host cell proteins were detected by the F550 antibodies for a coverage of 97 to 98%. 1,639 host cell proteins were detected by the F550-1 antibodies for a coverage of 97%. The coverage of these two antibodies agrees with one another. AEMS was used to determine the equivalency of the F550 and F550-1 antibodies. As shown here, the two CHO antibodies are 96% similar to one another as determined by AEMS. The Venn diagram below shows that 1,614 proteins were matching, as shown in green. The 35 unique proteins were identified by the F550 antibody in blue alone, and 25 unique proteins were detected by the F550-1 antibodies as shown in orange. By comparing the antibody coverage by AEMS, the F550 CHO3G antibodies have been successfully bridged to the F550-1 antibodies with the CHO antigen. In this example, we use orthogonal methods, AAE, 2D page, and mass spectrometry to determine antibody coverage for a HEK293 gene therapy bioprocess. The steps in this process were: Step 1, working cell bank; Step 2, harvest supernatant; Step 3, end of production cells; and Step 4, final drug substance. F650S ELISA antibody coverage of 60 to 90% was seen through steps 1 through 3. However, in the final gene therapy drug substance, the sample volume was limited, and the total protein concentration was far too low for 2D page. Gene therapy final drug substances are an ideal scenario for using mass spectrometry. AEMS was performed on the final drug substance, and the antibody coverage of the F650S ELISA antibody was determined to be 80 to 89%, with the identification of 235 proteins. AEMS is well suited for determining antibody reactivity to host cell proteins in the final gene therapy drug substance. For the next section of my presentation, I will present case studies where AE and MS were used to determine host cell protein antibody coverage to identify and quantify host cell proteins in drug substances and in process samples. In addition to harvests, we also analyze drug substance material for co-purified host cell proteins by antibody affinity extraction. In this workflow, an aliquot of a drug substance sample is saved. Then the remainder of the sample is run over the AAE column. The immunoreactive host cell proteins are captured with the immobilized polyclonal ELISA antibodies and are eluted from the column. Pre and post AAE samples are analyzed by LCMS analysis, and the results are reported as a list of all the identified host cell proteins, the drug substance, the host cell proteins reactive with the ELISA antibodies, the host cell protein molecular weight and isolation points, and the percent antibody coverage. This slide demonstrates the power of the AEMS approach for a host cell protein identification in the drug substance. The relative abundance of the drug substance and host cell proteins according to the extracted ion chromatograms were normalized to 1 and graphed in a stacked bar chart. Pre-AE on the left shows the drug substance heavy chain in orange and the drug substance light chain in light blue. The host cell proteins are beneath the two major blocks and are squished in different colors below. Post-AE on the right shows that the relative abundance of the host cell proteins has been increased after AAE, and that of the drug substance heavy chain and light chain has decreased. This slide shows that AAE enriches host cell proteins and depletes the drug substance. AAE is a very useful technique for determining that the ELISA is fit for purpose. This is because it can be used to show antibody coverage in the drug substance, the most important step. AAE is unique compared to other antibody coverage methods because it can enrich host cell proteins above the limit of detection of the MS and deplete the drug substance. In the top panel, we show a virtual 2D gel of CHO host cell proteins in a monoclonal antibody drug substance. The drug substance proteins are shown in orange. The matching host cell proteins are shown in green, and the AE-enriched host cell proteins are shown in black. Mass spectrometry is a very sensitive technique and can accurately identify host cell proteins in the presence of drug substance. AAE enriches host cell proteins in the presence of drug substance, allowing for their identification. In the bottom table, we see the number of host cell proteins identified in 4 different bioprocesses. Mass spectrometry identified host cell proteins, and AEMS was able to identify host cell proteins below the mass spectrometer limit of detection. The coverage for all 4 independent drug substances was 100%. This shows that Cygnus Technologies antibodies are developed to quantify host cell proteins in the final stage of drug substance purification. As shown earlier, AAE enables detection of otherwise obscured host cell proteins. This is important because without enrichment by AAE, mass spectrometry alone is not sensitive enough to detect and quantify known problematic CHO host cell proteins. An upstream CHO bioprocess sample containing drug product was analyzed with and without AE by mass spectrometry. The bars in blue represent the pre-AE sample, and the bars in red represent the post-AE sample. ppm quantification was calculated relative to the most abundant protein present in each sample. For the pre-AE sample, known problematic CHO host cell proteins are quantified relative to the drug product. For the post-AE sample, CHO host cell proteins are quantified relative to the most abundant protein, CC motif chemokine 2. By comparing the pre- and post-AE samples, the data shows several things. First, there is a roughly 50% decrease in drug product by AE. Second, and most importantly, there is detection of previously undetected known problematic CHO host cell proteins such as dipeptidylpeptidase 2, serine protease HRTA1, and plasminogen activator inhibitor 1. Thirdly, there is not a stepwise decrease in the ppm of each CHO host cell protein, meaning that their abundance has changed after AAE, potentially due to the drug product obscuring their detection. AAE enriches known problematic host cell proteins for mass spectrometry detection above the limit of detection. Next, I'll present how AEMS can be used as a tool to conduct investigations during manufacturing scale-up and transfer. A customer had developed a purification process at 50 liters and 450 liters. However, when they scaled up to a 2,000-liter GMP process, the levels of host cell proteins increased fivefold as seen in Figure A. Upon investigation, mass spec and ELISA quantification agreed that the increase in total host cell protein concentration occurred from step 3 to drug substance. AAE-MS was used to identify and quantify host cell proteins that were not cleared during purification steps 1 through 3 of the 2,000-liter GMP process. These host cell proteins shared similar molecular weights of approximately 100 kilodalton and isoelectric points of 5 to 8. One host cell protein, SNX9, was enriched eightfold from steps 2, 3, and drug substance and has a molecular weight of 66 kilodaltons and an isoelectric point of 5.2. SNX9 is located at the cytoplasmic vesicle membrane and is involved in endocytosis and intracellular protein transport. Mass spectrometry data can provide biochemical information about which host cell proteins are co-purifying with drug substances. These data provide actionable insights for process development groups to tweak the downstream process. A customer had developed a purification process for the drug substance that had host cell protein concentrations within their specification. However, when the process was transferred to a different GMP CDMO, the host cell protein level exceeded their specifications. AEMS was formed on the process samples from clarified harvest, step 1, step 2, and drug substance as shown in Figure A. A single 57 kilodalton host cell protein with a 5.3 isoelectric point was quantified in GMP run only. Using the host cell protein biochemical information that AEMS delivered, the GMP run was refined 3 times by the bioprocessing groups, with GMP runs 2 through 4 as shown in Figure B. The data shows that GMP2 decreased the host cell protein X level, but GMP3 increased the level of host cell protein X. The process group ran GMP4, and in host cell protein X was shown to be reduced to non-detectable levels by AEMS. This is a clear demonstration of how AEMS can guide downstream process improvements. I thank you for your time and attention, and I await your questions.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what is the sample volume requirement for coverage analysis by AAE with 2D page and AAE-MS?
Oh yeah, this is a good question. We get it quite commonly. So for 2D page and MS, if you submit a harvest sample, we typically like a sample volume of 20 to 40 milliliters. For drug substances, this is typically at least 50 milliliters.
And what is the protein concentration requirement for AAE-MS?
Yes. So this is a little bit similar question to the first one. So we like to do AAE, we like a concentration about 2 mg per mil and 20 mils. However, for AEMS, this works out to be the actual requirement is you want about 100 micrograms total of host cell protein.
All right, and our next question is, do you provide top 40 problematic HCPs for each of the cell lines you analyze?
Yes. So we've been doing host cell protein analysis for over 25 years now, and through those projects, we have learned that there are potentially problematic host cell proteins in each of the expression systems. So top common expression systems being um, CHO, E. coli, followed up by SF9, and then HEK. So for each project, we will generate a list of top 40 proteins. So these are from our years of experience as well as um, the literature. So over 20 years of host cell protein literature, and then um, any orthologs. If there's a human ortholog to a CHO ortholog, we will provide that information in the top 40 list.
And how do you define the top 40 problematic HCPs?
So that's I think on slide 2 or 3 I presented potentially problematic CHO host cell proteins. So they can be from causing drug instability, immunogenicity issues, degrading drug stabilizers, any host cell proteins that cause dilutional linearity issues. There's not enough antibody excess. And so those are functional reasons. The other way is just empirically and from the literature. That's how we define that list.
All right, great. Well, it looks like we're just running a bit short on time, so if we don't get to your questions, we'll be passing them along to Jared, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what about those proteins that are not recognized by anti-HCP antibody?
So when you run a gel, you'll just have spots. You won't know what those host cell proteins are. That's why you should run AAEMS, so you'll actually know what those host cell proteins are that may be non-immunoreactive to your ELISA host cell proteins. So in that case, you could go 2 ways. The first way is you should, if it's a generic antibody, a platform antibody, you should think about going and maybe into a custom antibody, which Cygnus can develop, and that may increase your coverage of those proteins that are missed by the platform antibody. The second thing is, if you have mass spec data, you can identify what those host cell proteins are, and then you could do a risk analysis. Are these proteins on the top 40 list? Are they potentially problematic? And then you could do a risk analysis and go from there.
All right, well, thank you so much, Jared.
Thank you very much.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.